Hello and welcome. In this video, I'll walk you through how to use the FunARTS web server, a tool designed to support target-directed genome mining in fungi. FunARTS, short for Fungal Antibiotic Resistance Target Seeker, helps researchers identify core genes, duplicated genes, and known resistance targets that are located near biosynthetic gene clusters, or BGCs. These associations can provide important clues about how a bioactive compound works or how the organism protects itself from its own toxins. The platform is intuitive and easy to use, and it automates a number of complex analyses that would otherwise require several tools and steps. Let's get started by uploading some data. On the main page, click on the Analyze tab to open the job submission form. You can upload either a GenBank genome file or a JSON output file from Antismash. If you're using a GenBank file, just click on Choose Files and select your annotated genome. Here I'm using a file for Aspergillus nigricans, and then hit Uploads. Your file will appear in the list of uploaded genomes. To ensure good results, it's important that your file is well annotated. Incomplete or messy annotations can reduce accuracy. If you already have Antismash results, you can instead upload the JSON output file. FunARTS currently supports JSON files from Antismash version 6 and 7. In this case, all the biosynthetic gene cluster information will be automatically integrated into the analysis. Once your data is uploaded, you can optionally enter an email address to get notified when your job is finished. That's especially handy for longer runs. You can also give your job a short title, like the strain or a project code, just to keep track of it more easily. Next, you'll need to choose the appropriate reference set for your genome. FunARTS includes curated core genes and resistance gene sets for different fungal groups. Picking the right one helps ensure more accurate results. Since our example genome belongs to the Ascomycota, I'll select that as a reference. If you're unsure which group to pick, you can always go with others as a fallback. Below that, you can choose which analysis to run. The core analysis, which looks at duplication and BGC proximity, is selected by default. You can also choose to scan for homologs of known resistance targets or search for domains of unknown function, or DUFs. While DUFs don't tell you much directly, they might be interesting for exploratory purposes. There's also an exploration mode that skips filtering to show a broader set of possible hits. Just keep in mind that it may include more false positives. And if you'd like to access the result link later without entering your email, you can choose to save the link in your browser cookies. For advanced users, there's an option to upload your own custom resistance or core gene models in HMM format, giving you more control over the analysis if you're working with unusual or poorly characterized organisms. Once everything is set up, click Submit Job to launch the analysis. You'll be taken to a status page where you can follow the progress. At the top, you'll see the job status, queued, running, or done, and there's a progress bar that shows how far along the analysis is. You can also open the log to see more technical details about what's happening behind the scenes. When the analysis is complete, the results are shown right on the same page. At the top, there's a summary section that gives you an overview. How many genes were found, how many core genes, and how many BGCs. Below that, you'll see a detailed table of all identified core genes. For each one, you get functional annotations, duplication status, links to nearby BGCs, and matches to resistance models, if any. The final 3 columns highlight key FunARTS criteria, and we can say that genes with duplication and proximity features are considered promising candidates. In our task, you get 2 proteins that fulfill these 2 FunARTS criteria, with a hit for known resistance gene. So, these core genes would be strong candidates. You can expand each row using plus buttons to see additional details. Here you now see the sequence IDs FunARTS gives to each gene, duplicated genes, if available, then the contig and the location of the gene. If a gene is associated with a BGC, you'll be able to click through to the cluster page in the BGC proximity table directly. There's also a separate section showing known resistance genes that were detected in the genome. These can point to potential self-resistance mechanisms or give you clues about how compounds might work. Further down, you'll find duplication results for the core genes, which can highlight genes that may have been amplified for functional reasons. The BGC proximity table displays all the clusters detected in the genome within the anti-smash view. Additionally, you can expand each row to access detailed information about gene types, locations, and functional annotations. For example, we can display one of the promising candidates in our genome. It is R5C8 and beta-lactone, the main biosynthetic genes. The main biosynthetic genes are indicated in red, while core genes are shown in yellow. A core gene that aligns with a known resistance model is labeled core res and appears in purple. Conversely, a gene that is solely recognized as a resistance gene is displayed in light green. Finally, all results can be downloaded as a zip file, so you can review them offline or use them for further downstream analysis. We have examined how to analyze a single genome file. Now, let us review step by step how to analyze multiple genomes in the same job. To do this, we're coming back to the Analyze page. Just like in the first section, we prepare our genome files. The only difference is that this time we will upload multiple files at the same time. During file uploading, you can click the Choose File button and select multiple files at once. For example, here we select 2 genome files and then click the Upload button. Alternatively, you can upload the files one by one. Again, we click the Choose File button, select a genome, and then click Upload. We repeat the same process for the other genome file. In this example, we showed how to upload 4 different genomes. Once the files are successfully uploaded, you will see all the genomes listed in the Uploaded Genome box. The next steps are exactly the same as for a single analysis. We select the reference set and analysis options and then click Submit Job to start the analysis. This process directs us to a special page showing the results for multiple genomes. Although the page layout looks slightly different from the single analysis page, the information it contains is a combination of individual results. You may need to refresh the page to see updates as the analysis progresses. At the top of the page, there is a table containing summary information for each genome analysis. This table shows the number of FunARTS hits from individual results. You can also see which genome analyses have been completed, and clicking on the relevant row will take you to the detailed result page for that genome. We have already shown how these results are examined in the first section. If you wish, you can go back and review it again. The table is automatically updated once all analyses are complete. If the update does not occur, it is recommended to manually refresh the page. Our process is complete, and now we can take a closer look at our results. After the summary table, we can look at the core gene table. The core gene table displays a table similar to the single genome view and summarizes the data for all uploaded strains. It is important to note that the figures under duplication, BGC proximity, and known resistance hit represent the frequency of hits found in total input number. A score of 0.0 indicates that no hits were found for any of the FunARTS criteria in any genome, while a score of 1.0 indicates that the criterion was detected in all uploaded strains. The next 2 tables are resistance models and duplication tables. The number in these tables represent how many genomes have the corresponding hit. Bigscape result of the given inputs are also represented at the bottom of the results page. Here, the clustered BGCs from anti-smash results are shown with associated core genes and resistance models. Open Bigscape Overview option also allows visualization of the interactive Bigscape result page of the clusters. Finally, you can export all the tables and alignments on the page. You can also easily download the zip file containing all the analysis results by clicking the button at the bottom of the results page. Remember that your FunARTS results are stored on the server for at least 1 month. This means you can access them again later. And that's it. You've now seen how to upload data, configure the analysis, monitor its progress, and interpret the results using the FunARTS web server. Whether you're trying to discover new bioactive compounds, understand resistance mechanisms, or just explore the biosynthetic potential of a fungal genome, FunARTS is here to make the process faster and easier. Thanks for watching, and happy genome mining!